Hello everyone, I welcome you to another series of Mendel's bicentenary celebrations. I'm glad to introduce myself. I am Mercy Rofina, pursuing my PhD in Dr. Vinod Skaria's lab at CSIR IJIB. So today we have with us Dr. Giriraj Chandak, who is a chief scientist from the Department of Genomics and Proteomics Research at CSIR CCMB. He's here to discuss with us his opinions about Mendelian discoveries, human genetics, and how it is paving a way for modern research in science on a current scenario. His research group mainly focuses on understanding the genetic basis of complex disorders like diabetes mellitus, chronic pancreatitis, and so on. So it's our pleasure to have you here in this platform today, sir. Thanks for joining. And it would be great if you could elaborate us on what exactly your current research interests are. We already knew you're working on some complex genetic disorders, and it would be great if you could share with us what your lab is currently doing and what you're progressing forward.
Okay, thank you for having me on this important occasion. It's my privilege to be sharing my experience of last 2.5 decades or so. And so I'm actually trained as a physician. So for me, Mendel and the knowledge about Mendelian genetics was very primitive, when I decided to move on to this field. But this is one of such things that you cannot really do genetics without knowing about this. So I must say that I learned it while conducting my research on this. But with the basic knowledge of Mendelian genetics during our medical curriculum, I could focus my research in a very systematic manner, I would say. I would just tell you some examples. How did I move forward and where at each step Mendelian genetics or a knowledge of Mendelian genetics, helped me. So when I came to this field, I started working on largely the simple genetic disorders like sickle cell anemia, thalassemias, and others. And there the idea was that these are all Mendelian genetic disorders. I'm talking about 2 to 3 decades back when most of the disorders were considered as single gene disorders. Okay. So at that time, I was largely, being a physician, I was largely focusing on the knowledge on genetics of these diseases, understand this better, and then provide genetic testing, prenatal diagnosis, and counseling to the large bunch of patients. While doing this work, I came more close to understanding the clinical phenotype. And as I said from my earlier MBBS knowledge also, I had a bit of idea that the clinical presentation of many of the disorders is not similar in this country compared to people abroad. So somehow that became the focus of my research, so which I called as transition from simple to complex genetic disorders. And I think probably I was largely contemporary at that time because that's the direction that the world was also taking. So slowly I moved on to understanding the complexity of these single gene disorders. I worked on sickle cell anemia, and then slowly we found that the genetic basis of why the phenotypic heterogeneity is there in sickle cell anemia. I talked specifically about sickle cell because that is one single mutation which is globally associated with sickle cell anemia. Yet we found that the disease is so mild in Indians compared to others. So I'm not going to go into the details, but then we did quite a bit of work on that. And that actually focused my attention more towards understanding the complexity in these kind of disorders. So we moved on to complex disorders. And one more factor which was driving the research on other complex disorders, because sickle cell anemia is still, one of the rare disorders also. So we started our focus on diseases which were of high prevalence. So we worked on pancreatitis, worked on diabetes, and many other disorders. And the idea again was to look at that why there is a phenotypic difference in the presentation in Indians, and whether this is all because of the genetic susceptibility. And the last part, which I just started doing in my last 10 years or so, is that so we were all bitten by genetics bug. But then we realized that it's not genetics, that's the hammer for all kind of problems. And then we understood that largely genetics would probably be similar across populations. There might be mild differences, but then there may be other links like environment, or I'm using environment very loosely, but this is the most complex thing to understand. So I would summarize by saying that currently my lab's focus is to understand the genetic susceptibility of complex disorders, and understand that how the genetic susceptibility is influenced by other factors, which can be your lifestyle, environment, and finally, how can these risks be modified? Because that genetic risk is largely unmodifiable, particularly for complex diseases.
Mhm.
Yeah, so that's what my group is currently doing largely.
Right. Thanks a lot, sir. It was really indeed very, very interesting. And so this brings me to my next question. Since you stated that you saw a transition from being a clinician, coming to science and understanding the field. So right from the time you started to the current scenario, how do you think the translational effect of science, basically genetic science, has developed? Is it having a more impact now, or how do you see the transition in all these years, right from the time you started?
Yeah, so I would say that the field has really blossomed, very heavily.
Mm-hmm.
Since I work in, India,
Mm-hmm.
so I would largely focus my, impressions from my research, and then my colleagues and others research, and try to compare it with how things have been happening all over. I
Mm-hmm.
must say that the human geneticists and other people in the country are largely contemporary, and the kind of research that takes place is really exemplary. So I have been privileged to have many of the colleagues around with whom one could talk, and get more knowledge and approaches to follow while dissecting this complex problem. I agree with you that now, slowly over a period of time, as the knowledge about the genetics of diseases has improved quite a bit, and
Mm-hmm.
more importantly, the sort of genotype phenotype correlation, which means that uh, how does the genetic heterogeneity influences the phenotypic heterogeneity. And
Right.
lastly, as I said, that what are the other factors that can probably influence the genetic susceptibility.
Mm-hmm.
So overall, since I started, it was largely, I had the privilege of starting one of the first sort of centers for genetic diagnostics. So at that time, the translation largely was if we were able to identify the genetic defect
Mm-hmm.
and be able to do a prenatal diagnosis, genetic counseling also.
Right.
But we know today that anything and everything that you talk about the phenotype, right from, as I said, susceptibility to clinical presentation to complications to treatment, everything seems to be guided by genetics. Um, uh, so I would say that as the knowledge has kept on increasing, this all has gone into the foundations of translational perspectives. I'll
Mm-hmm.
just give one example. Like when we started with chronic pancreatitis about 2 decades back, the
Mm-hmm.
only motivation was that the disease was presenting very early in life, and then it was very severe in Indians. And it was called as tropical calcific pancreatitis. It was a historical thing coming from south. And so our only motivation was that, well, if genotype drives the phenotype,
Mm-hmm.
then what is it that is different? And to
Right.
cut a long story short, we found that there are the genes that are responsible for the disease abroad, they are not so in Indians. And actually, we found two novel genes which we're doing. So I would say that in such kind of investigations, when we started, it wasn't with any motivation of doing any translation or whatever. But then after a few years, we realized that this can actually be used as for a diagnostic or a screening testing for any Indian patient with tropical calcific pancreatitis. So the point I'm trying to make is that at that point of time, people were largely looking at understanding these questions, the differences. The translation was probably not so much in mind. But then today, I think that, most of the research is getting focused with an aim to get something back to the patients in terms of translation. We can use a broad term of translation.
Right, right. Indeed, very true, sir. So you have stated that the prevalence of the disease is varying, like when you compare it with India, with other Western countries. So do you think a subpopulation is very specific, like a particular geographical population? In that case, what is your comment regarding the stigma that is attached to most of the genetic disorders? So there's always a stigma coming along with the fact that it's running in the families or it's running in the communities. Have you ever come in contact with any patient families, or do you have any experience regarding that?
Oh my God! I mean, that has been the most difficult part of my career. When I was in the beginning, and that is what I mean. I always used to tell myself that as physicians, we are not trained in these things. We are always trained in treating patients in our cozy clinics. But when you take your clinic out to the population, then these situations come up. I'll just share one or two experiences. When at that time there wasn't much awareness about these diseases, so it was very difficult to expect that the patients would come for this kind of testing. So I decided to tag along various NGOs and patient support groups. So there's Indian Association of Muscular Dystrophy, which interacts or rather takes care of muscular dystrophy patients. Then there was Hemophilia Federation of India, which was all about hemophilias also. So I decided to get in contact with these because they had well characterized patients, and I thought that will be the best way to help the patient and through them create the awareness about diseases. So I mean, my first visit itself was an eye opener because we went for a camp, and so it was for hemophilia actually, and I wouldn't see any female. It was only the male and the father who were there in the whole thing. And as you are aware that this is an excellent recessive disease, which means that females are obligate carriers and males are affected. So it was a shock to me that the whole of the effort on counseling and other things will be a waste unless we are able to create awareness about that. And secondly, that it also showed the male dominated society, which had already put the blame on the females, so much so that they didn't even think appropriate that the females would really know about it. And I'm reminded of many instances where a family has come, the lady has come during her pregnancy stage to our institute at CCMB, hiding from her family, from her husband, for doing the testing, sampling. And one time I will never forget that the husband and the husband's family, they followed the lady till CCMB, and we had to shut the lady in a room saying that it is a group leader's room and it cannot be opened. So these are really, unforgettable experiences. But good part is that, things have really changed now. I must say that, the clinicians and community and overall, I would say through government approaches also, this has helped quite a bit.
These are like really very, very unique examples. Like we would have heard it in this series of interviews, sir. So that was really interesting. And sir, you have seen a lot of faces being in this field, and now you see genomics getting into connect with genetics. All of the interdisciplinary aspects are coming together. There's a fall in the sequencing techniques also. So what do you think will be the future advancement in genetics or genomics, which will help in shaping the health policies of nations in a better way in future.
Right. So I see, I'm not a big fan of, technology, to be honest. I like technology till the time it comes to the societal benefits, or, asking or answering a very good research question.
Mhm.
Clearly, what you said is very true, that, the burgeoning of these technological advances has made it possible now to basically, probe each and every base of
Mhm.
the whole genome.
Right.
But at the same time, this is my personal,
Mhm.
impression, that I feel that, while this is a tool
Mhm.
for, like getting to know about each base of the genome of a patient, or any individual, but at the same time, we really don't have the know-how to understand that what they actually mean.
Mhm.
So if I talk about complex disease genetics also, there are a large number of genetic loci which are known to be associated,
Mhm.
so the conversion of association to causality, that is something which I think should really be, looked at, because we
Right.
are all sitting on heaps of these kind of associated variants. And
Mhm.
same thing is happening to, monogenic disorders also. So I would say that this has become a tool in the hands of clinicians, as simple as, writing a prescription for glucose testing or something. I wouldn't say that they are not aware, but I guess that is the best tool at this point of time. So I would surely think that, there should be now advancements in understanding that what do these variants do, and that I think really needs a very, very interdisciplinary approach. That, there is a big, it can be a dream situation or whatever, but then say you are in a setup where there are clinicians well aware of the genetics. They look at the phenotype, very well characterized, get them investigated, get a proper genetic testing done. And there is a set of, basic biologists who can, quickly look at using either cell lines or looking animal models or whatever. Although these are easier said than done, but I think that this is some sort of an approach which is, required to be, tested. Yeah, I think that's what will be very good. One more point I would make is that, I realize, this is my personal experience, that it's like when you get into a little high fancy car, you just forget ambassador or something, which has taken us for decades, being very sturdy and all that. So I find that most of the focus, or rather the fancy or fashion, has become to prescribe exome sequencing,
Mhm.
whole genome sequencing. And to me, I'm not blaming anybody, but to me it seems that the other, I would say, relatively commonly occurring rare diseases like thalassemias, muscular dystrophies and others,
Mhm.
I somehow feel that we hear less about, those, because they have become like a glucose test, or a urine test or something. So I would say that, in terms of government policies, while it's good to always focus research and all these, but I would be really very, very happy if there are focused programs around, some of these diseases like hemoglobinopathies, musculopathies, for which we know the real test required, and possibly there are likely treatments also possible. So the model that I suggested, earlier, I think that can be cut short because you have a knowledge of genetics, and that can immediately take one to translational perspective. So I hope that government, thinks of putting focused programs on these sort of commonly occurring rare diseases and rarely occurring rare diseases. Yeah.
Right, right. Very, very correctly stated, sir. So, from whatever you have told, one point to the people who are currently in genetics for young researchers is like, once you have data in hand, like we are supposed to think in different ways, in an interdisciplinary way, to know more about the basic biology, the role of the variants, rather than just keeping the data in hand. We need to drive more further towards understanding its basic roles. So that was a really motivating point, sir. So this brings us to the end of our discussion. So if you have any take-home message, you've already stated in your earlier responses for young researchers who are starting to enter genetics or who are already working on genetics, if you have a kind of motivation pill or encouragement message for them to move forward in this field, we would like to hear that from you.
Well, I must say that, it's difficult to, say any message for anybody. I mean, I'm still learning, so it's like, learning messages from each other. But I mean, I would definitely say to this human genetics community, young, old, whatever, from my experience, I would say that it's very, very important to, keep the eyes open and have mutual respect for every colleague, because as I tried to hint that, still there isn't that sort of collaborations in the field of human genetics, particularly, with reference to the, I would say, the patient benefit or the societal benefit. I mean, it's a little sad, but I think that as we have evolved in last 2 decades, we would evolve further. But I think that that is where the young researchers, with their very open approach, and I actually consider them as the knowledge tank and the think tank. They can drive many of the ideas. I mean, I must say I'm privileged to have so many young people in my lab, and
Right.
now most of my research is guided through them, because the field is so huge also. And I would definitely say two more things. One is that uh, try to understand the phenotype as best as possible, because these technological advances, they will only let you have the data. You can keep a mountain like anything, and you can keep sitting happily on that mountain of variants of unknown significance or patients on whom you don't have a diagnosis. But unless you really, like say, once the genetic test is done, again get back to clinician, because clinician might like to do some more tests or other things. So I think that's very, very important. And also my request to, basic scientists and others is also that, uh, don't consider clinicians or anybody just as the blood sample provider. They are very, very valuable. It used to be earlier, I guess, but now it's extremely important that there is a one-to-one interaction and a regular interaction on a long-term basis. I think that can be one of the ways that the science can actually reach the society and make a difference in the health of the people. Yeah. Thank you. Yeah.
Thanks a lot, sir. Thanks a lot for taking your time and sharing your thought-provoking ideas, opinions, and your views on the current science which is being done in the nation. Thanks a lot for joining us today, sir.
Thank you very much for having me. It was my privilege. All the best.
Thank you, sir.
Bye.
Bye-bye.